Hello, good afternoon, everyone. We are going to finish lecture 4 today. And we'll see if we have time to talk about the lab. If not, we're going to wait until next week to do so. Let's go ahead and get started. So last class, we talked about membrane structures in greater details with the fluid mosaic model. And we also explained the different functions related to membrane proteins. There are 6 functions that we talk about specifically. And one of the 6 functions of membrane protein is the fact that they participate in transport. They're able to help move things across the cell membrane in some cases. And that's what today's lesson is going to be about, the types of transport. So the most straightforward way things tend to move around in the environment is by using what's called diffusion. In diffusion, molecules tend to move from an area of high concentration to an area of low concentration. The best way to visualize this is to imagine you have a flask of water and you add some dye to it. Okay, so this area is high concentration, and we can put square bracket to represent concentration. That's like an official way of how you do it. And elsewhere in the flask, you will have low concentration. So even if you don't stir it or swirl it or touch it in any way, the dye will automatically move and spread across the entire flask. Right? You come back to it like the next day, the whole flask is going to be blue, right? So this natural migration of the dye from a concentrated area to less concentrated area spontaneously, this is called diffusion. Right? Another example is if you spray some air freshener in one side of the of a room, right? Soon you'll be able to smell it across the room, right? The molecule naturally diffuses down their concentration gradient. Right? It goes from high to low concentration. That's called diffusion. Now there are different factors that affects the rate of diffusion. The rate of diffusion is talking about how fast is it happening. Okay. If it spread really quickly, that's a high rate of diffusion. If it spreads slowly, then there's a low rate of diffusion. Okay. So factor number 1, size of the molecule. If it's smaller, it tends to spread faster. So if you think of something like over here, we have O2 in this picture, and this is like C6, H12, O6. Something big like glucose is probably going to take some time to diffuse compared to something smaller like oxygen. Temperature also plays a role in terms of diffusion. When it's higher temperature, they tend to spread faster. Okay. An example is like on a hot summer day, sometimes if it's garbage day, you can smell the garbage like really, really easily, right? Because it's hot. But on a cold winter day, you barely smell anything. Okay, like the molecules associated with odor, they're not really spreading up. Right? It's very slow rate of diffusion when it's cold. Next factor is called the steepness of concentration gradient. Okay, a concentration gradient is just talking about the difference in concentration between 2 points. How much of a difference are we talking about? The greater the difference, the steeper the gradient, and the faster the rate of diffusion is going to happen. Imagine you have a ramp. Okay, like you put a ball down versus if you have another ramp like this. Like which one is going to roll down faster? Well, the steeper one is going to go down faster, right? Because there's a greater difference in height. So similarly, when we talk about concentration, like the top picture here, I'm making this number up, okay? Let's say this is 80% concentrated. This is 0%. So we have a difference of 80. These red circles really want to go over there. Okay, it's going to be going there really fast. Over here, let's say we have a 60% and this is 10%. So the difference, I mean, there is a difference. It will still move, but it's not going to move as quickly. Okay, so they'll still go, but it's going to take some time. Right, so the greater the difference between the 2 points, the steeper the concentration gradient, and the faster the rate of diffusion is going to be able to occur. Does anybody have any questions so far about the size, the temperature, and the steepness of the concentration gradient before I tell you about the last one? Okay, the last one is a little bit difficult to explain, over the internet. I'll try to see if it makes sense here. Give me a moment to see if this works. Oh, okay. So let me just move all these things over here to one side. Okay. Move all of them to one side. All right. So naturally, things are gonna go from the left side here to the right side, right? Because there is not a lot. There's nothing here, and then there's a bunch of H positive here. Okay. So diffusion, high concentration to low concentration. But what makes it different here is that they are all positively charged, right? What happens if you put a positive charge next to another positive charge? Does anybody know what I'm getting at? If you have two...
They escape from each other.
That's right. They escape from each other. They try to move away, right? The term I was looking for is that they repel each other. Things that have same charge will repel each other. Two positives will try to get away from each other as far away as possible. Same thing for the negatives, right? So not only the diffusion is gonna drive them apart, but the fact that they are both positive, the fact that they're all positive, sorry, means that they will try to get away from each other as quickly as possible. So at the beginning, you're gonna have really fast diffusion. Okay, just try to get away. However, as they move to the other side, as the positive charge builds up on the other side, they are gonna push back a little bit, right? Like this next guy is gonna have a bit of trouble. It's gonna try to go over, but then it will feel the positive force pushing back, right? So like it's not really going. Oh, you can't really see what I'm doing on the screen. Sorry, I just realized that. Let me try that again. As this guy, right? This guy tried to go over, and then it's gonna feel the positive kind of pushing back, right? And then it's gonna push back. So at the beginning, it's gonna increase the rate of diffusion, but as time goes on, right, there's gonna be some pushback, right? So the charge is gonna affect how fast the rate of diffusion is gonna occur. Does that make sense, that last part there? Anybody wanna ask anything? Okay.
Means that if the environment, is there any possibility that environment that they diffuse is like having charge. Like if the H positive goes to the right side, if there is something there and comes back again, right?
That's right. So like as you will see, lecture 5, right? There are parts in the cell where some places are more positive than others. Okay. And that's going to affect how the things move. That's essentially what we're getting at. Okay. All right. Next, we're going to talk about this concept called osmosis, and I want to take a note with you. Okay. Lots of stuff to write today. Osmosis. So osmosis, the definition is the diffusion of water. Diffusion of water. Now, please pay attention to this question here because many people does not get it right on the quiz. So let's say I have a tube here. Okay, it's a U-shaped tube. All right. And I am going to put a membrane here. This is a selectively permeable membrane. Only water can move through, can move across. Okay. So I'm gonna fill this up with water. I'm gonna fill it up with water. There we go. I hope I'm making sense here. And if I'm not, ask questions. I don't know why it is not letting me erase. Hold on, let me see. Maybe it will get bigger. Oh, there we go. Okay, so same water level, right? Same water level. Now I'm gonna dissolve some solute in here, okay? So solute. And then I'm gonna dissolve more here. Okay, these red balls, red circles. Excuse me. That is the solute. And the important thing to remember in this exercise is that they are too big to cross the membrane. Okay, you can see the way I've drawn the membrane, right? Like the gaps are really, really tiny. The red balls are too big. They're not going to be able to cross. Okay, so the only thing that can move in this situation is the water. Okay. My question to you is, which way is the water going to move? To the right, to the left, or no movement at all? Let me launch the poll and see what people think. The question is, which way will water move? Okay, very good. Anyone else? Last call? Okay. So we have A and C being the popular answer, and then some of you chose B. So let's take a closer look at this, right? Okay. Right now, do you see that the solution on the right has higher concentration? Because it has like more dots in the same volume, right? And on the other side here, we have lower concentration. Okay. If this was like a salt solution, you taste the right side, it's gonna be saltier than the left side, right? I just want to make sure that you understand that first. So what happens? Well, the red dots, the solute, okay, the salt, for example, the red dots, they really want to move to the left side, don't they? Because they have a tendency to go from high concentration to low concentration. But they can't because the barrier won't let them go through, right? So what's going to happen? Well, since the red dots cannot move, so the only thing that can move in this case is going to be the water, right? And this whole concept is about balancing things out. So if it's more concentrated on the right side and less concentrated on the left side, the water will actually move to the right. Right? By moving to the right, the water will dilute the solution on the right. And at the same time, you will be concentrating the solution on the left. Right? So the water level drops on the left, but the water level will rise on the right. And that's the direction of water movement. Okay? So afterwards, this will continue until the concentrations are the same on both sides. Okay? Until the concentrations are the same on both sides. Okay? So water moves to the right to dilute the solution on the right. Okay? While concentrating the solution on the left. And this will continue to happen until the concentration balances out. Okay? Remember, the red dots did not move. It's only the water that moves.
Excuse me, Professor. I didn't understand why the water moved to the right, because of the more dots there.
Yeah, because there are more dots there and the red dots cannot move, so it sucks the water to the right side. Here's a way to help you guys visualize this. Then you can try this too if you want. Okay. If you take a spoon of salt and you put it in your mouth and you hold it. Okay, keep it there. What's gonna happen? Think about it. What's gonna happen? Don't swallow, just keep it there for like, 15 seconds, 20 seconds. What's gonna happen? You're gonna drool, right? You're gonna feel the saliva coming out, right? That's exactly what's happening in this exercise here. The red dots, the salt, is sucking the water out from the other side, right? The saltier side is always pulling water towards that area, just like the spoon of salt is sucking all the moisture out of your mouth. Does that make sense?
Yes, Professor, thank you.
Yeah. Any other question? Okay, this question is gonna show up sometime. Okay, I don't remember if it's the quiz or if it's the test, but people always have trouble with it. But hopefully, after I explain it, you have a better idea. And watch the recordings again if you need to hear the explanation one more time. Okay, so this is the exact same picture. Remember, the red dots are not going anywhere. It's just the water level that's changing. Done. So this technique is useful for like dehydrating food. Before they invent refrigeration, people preserve their food by salting it, right? So if you add a lot of salt to meat, they become dehydrated. Without the moisture, the bacteria don't grow, right? And you can keep the food for longer. Okay, so we have another class note here. It's called tonicity. Okay, this is a long note. Tonicity. Okay, what does it mean? Tonicity refers to the ability of a solution to make water move into or out of a cell by osmosis. Osmosis, again, is just the movement of water across a membrane. Okay. So I'm going to explain to you 3 scenarios. 3 scenarios. Just stop me anytime if you don't understand something. Don't wait until the end of the note, okay? Just ask the question as they pop into your mind. I guarantee you, if you have a question, someone else in the class probably has the same question. Okay, if you don't want to ask, just type it in the chat box. That's okay as well. First case is called isotonic. Isotonic. Okay. So let's draw it. Okay, so we have a cell here. Okay, I'm just gonna use an animal cell here as an example. Animal cell. And then we're gonna have some solute. And in this case, the concentration on the outside and inside are the same. I want to bring up a point, okay? Listen carefully for a second. Stop drawing. When we say concentration, we are not counting the dots, right? Okay. Like even though there are more dots outside of the cell compared to the inside, they can still have the same concentration. Okay? Because the volume is smaller in the cell, right? Do you guys understand what I mean? Right. Yes, there are more dots on
Yes.
the outside, but then the volume is also bigger, right? So like when you do the calculation, concentration is basically like amount of solute divided by volume. Okay, so if you have small number divided by small number, that could equal to a big number divided by a big number, right? Okay, so it's not about the dots. So in this case, this is animal cell, and isotonic means the concentration inside the cell equals to the concentration outside. Okay. I will draw a plant cell as well. Okay, plant cell. Plant cell, of course, has a cell wall. Okay. I'm not drawing the nucleus because it would be too much stuff. Okay, so this is plant cell. Give you a moment to catch up. Maybe give me a thumbs up when you guys are ready. Okay, I'll wait for a few more people. Tell me when you're ready. All right, thank you. So because the concentration are the same outside and inside, the water will move in, yes, but water will also move out. Right? And the rate that the water is moving in is same as the rate that the water is moving out. Right? So rate in equals to rate out. So no net water movement. Okay? Which means no change in volume. When I say rate in and rate out, I'm talking about the rate that water is coming into the cell and the rate that water is leaving the cell. People might find it useful to think of this analogy. Okay, let's say I have a basin, a bucket, and I have a tap, okay? I am going to turn on the tap and the water is going to come in. Okay. So obviously the water level is going to go up, right? But I am going to poke a hole here at the bottom. I'm going to poke a hole so that the water is leaking. Okay. If the rate at which the water goes into the basin equals the rate at which is leaving, what's going to happen to the water level? Is it going to go up, no change, or go down?
No change. No change.
No change. That's exactly what's happening. The water is coming into the cell, but at the same time they're leaving. They cancel each other out. No change in the volume. This is called isotonic. Isotonic. Okay. If you are an animal cell, you absolutely love isotonic. This is the best. This is the best environment that they want to be in. If you are a plant cell, it's like meh. Okay. I don't love it, but it's okay. I'll survive. Okay.
Vincent,
Yes.
sorry, what's the no net H2 movement again?
Okay, so because the concentration of the red dots are the same outside and inside, right? So the water is coming into the cell, but at the same time, the water is leaving the cell as well. So if you have the same amount of water coming in as it is leaving, then there's no net effect, right? Net means like after you cancel everything out. Is that better or no?
Yes, thank you.
Okay, all right. So that's isotonic. Again, if it's an animal cell, fantastic, you love it. If it's a plant cell, they don't really like it. Okay, they prefer another kind of solution, but they'll live. It's not gonna die yet. All right. Let's move on to number 2. Number 2. Number 2, let's do hypertonic. Hypertonic. Okay, we're gonna do our animal cell again. This is a beaker, okay? It's just like a beaker of water, and you pop a cell in. Okay. I will draw the plant cell as well. All right, so this is the beginning. And in the case of hypertonic, you're gonna have a lot more solid. It's like highly concentrated on the outside, but not really on the inside. I hope you're using color, okay? If you're doing this in black and white, it's super difficult to you know, to distinguish what's going on. So in this case, the concentration on the inside of the cell is less than the concentration outside. And this is where I really need you to think about it. I think of the U-shaped tube that we talk about, right? Do you think there will be more water coming in this case, or more water leaving?
More water leaving.
That's exactly it, right? More water leaving, because the red dots can't cross the membrane. So, you know, the water is trying to leave to balance out. So you have a lot of water moving out. Does that mean there's no water coming in? No, it's still coming in, but you just have a lot more leaving is all. Okay? Leaving. So rate in is less than rate out. Net water loss. Okay, net water loss. Cell shrinks. Let's think about our water basin. Okay, I'm gonna turn on the tap. Water is gonna fill up, but we have a really big leak, okay? Like that, it's like, oh, going all over the place, the water. You're still filling it, but then you have more water leaving. So what's gonna happen to the water level?
Reduce.
That's right, it's reduced, it drops. So similarly, our cell volume is going to go down, it shrinks. Okay? So in an animal cell, the cell is gonna shrivel up like this. It's gonna shrivel up. Okay, this is called plasmolysis. Okay, not the cell is not happy. Okay, but it's okay. It'll live. Not gonna die. It'll be all right as soon as you rehydrate. But for a plant cell, For plant cell, the cell wall is gonna be intact. Okay, it's fine. But then the cell is like shriveling as well. Okay, it's also plasmolysis, and they don't like it. Okay, this is what happens if you forgot to water your plants for like a week or two. They literally shrivel up and, like, you know, become a little bit limpy and withering, right? That's what happens. Any questions before I tell you about the third case?
I have a question,
Mhm.
Vincent.
Yeah.
So, as you said, the plant cell become like shrinks. What about the animal cell? When they lose a lot of water,
That's
what
right.
will happen?
So this will shrink your cell, and actually it will, like, you have a part in the brain that when it shrinks, it sends a signal that makes you feel thirsty. Okay, so like that will cause you to drink water, right? And when you drink water, you will basically dilute the environment, right? It goes into your blood and it dilutes the environment, and then you will return to isotonic, right? But of course, if you ignore it and then, you know, the cell keep on shrinking and shrinking, there's going to be a point where you will be like severely dehydrated. Then that's going to be, you know, problematic, right?
Yeah, thanks.
Yeah. Okay. Number 3, last one. This is hypotonic. Okay, hypotonic with an O. And you guys can probably guess what this is like. And you're gonna have a lot more solute inside than outside. Same thing for our plant cell. All right, so in this case, the concentration of the inside is greater than the concentration outside. So again, I want you to think about how the water is going to be moving. Is it going to go into the cell more or out of the cell more?
Into the cell.
That's right. Thank you. So rate in is greater than rate out. Okay. So you have a lot of water coming in. Okay. It's opposite to what we had before, right? Before we have more water leaving. Make sure you get the direction of the arrowhead correct. So you have a lot of water coming in and not a lot leaving. So net water movement. What did I write before? Okay, I write net water loss. So this is net. I just wanted to keep the language similar, so it's not going to be too confusing. Net water in, as in like into the cell. Cell swells. Right? Before it shrinks, now it swells. Back to our basin. Hey, we're gonna turn on the tap, but we're gonna really turn on the tap, okay? Like turn it on to max, and then the water is leaving. Okay. So a lot of water coming in, but it's not really draining. So water level rises, right? And eventually it's gonna overflow. And it's gonna spill. And that's what's gonna happen with our animal cell. Okay. The animal cell will pop. Okay. It will explode. Okay. This is called lysis. Okay. Burst. Do we like this? Absolutely not. The cell died. There's no coming back from it. Right? Unlike the shrinking one, you can drink water and you can go back to normal. This one, it bursts, it dies, not coming back. You think the plant cell will burst as well?
No,
Why not?
The
because
cell wall.
That's right, because it has the cell wall, right? So the plant is absolutely loving it. It's gonna be like very, very puff, right? It's gonna take in all the water, and then the water is actually pushing against the cell wall, okay? And the plant is very happy because it has a lot of cell. Right? If you take a plant that's like, withered and then you just soak it in water, it will literally stand up and you can see the leaves are opening. This one is called turgor pressure. Okay. Missing an R here, sorry. Turgor pressure. So that's it. Are we together with these three scenarios? Hyper, hypo, iso. Oops, sorry. Okay. That's what these are red blood cell, by the way. Red blood cells. Okay, this is happy looking red blood cell in an isotonic environment. This is hypotonic. Okay, you can't tell whether it's burst or not, but if it ain't burst yet, it's probably gonna burst soon. And this is hypertonic when it's all like shrivel up like a raisin. Okay. If you have raisin at home, you can leave it in tap water overnight. Tomorrow it goes back to a grape. The water is gonna go into the raisin because there's a lot of sugar in there, right? So when you put a raisin into tap water, that essentially is a hypotonic solution. Okay. It will swell up again. So if you become a healthcare practitioner, you might have to deal with IV solutions. Okay, IV solutions is intravenous, right? Like you inject it into a vein, right? Depending on what kind of condition the person is experiencing, you will have to give them different kind of IV fluid. Okay, so for a healthy patient who just needs more fluid, then of course you're gonna give them isotonic fluid, right? So I'm just gonna show you a few different kinds of IV solution here, which you might encounter when you again become a nurse or some other health practitioner. So the 0.9%. You learn in chemistry, right? Oops. You learn in chemistry, salt is NaCl, right? So this one is 0.45 here, but I'm talking about the normal one. The normal one is 0.9%. There is a picture here, 0.9%. So 0.9% means there is 0.9 grams of NaCl per 100 mils of water. That's what it means. Right? So this is most common IV fluid. It's given for people who needs hydration. Okay. If they're bleeding, you want to give them some fluid. Vomiting, diarrhea, right? You're gonna give them the normal saline, isotonic. Right? The cell's gonna be happy. Right? We have a 0.45%. As you can see, it's half of the normal amount. So only 0.45 gram of salt per 100 mils of water. And this is for hypotonic. Okay, 0.5 this should say hypotonic. I don't know why this says normal. Hypotonic. Okay, so this is half normal saline. And this is useful to treat hypernatremia. Okay, hyper means too much. Too much, huh? Hyper is too much. Natri is sodium. You know the symbol for sodium is Na, right? That's because the Latin name for sodium is natrium. Okay, if you ever wonder why is it not So, it's because it's based on the Latin name. And so this is too much salt where emia. Right? Anytime you see emia is in the blood. So these are for people who are like severely dehydrated. Okay, it's the blood is too concentrated. So you want to give 0.45 to dilute it a little bit. There are people who are diabetic, they sometimes have these things called ketone that builds up in their body. And to kind of flush it out, to kind of dilute it, you also give hypotonic solution. Okay, when do you need hypertonic? This is 3%. Okay, it's really concentrated. This is really rare, okay? One example is to treat hyponatremia, okay? Water toxicity. Do you guys know like the video game console, like the Wii? This is really old, like, you know, it came out, I don't know, like 15 years ago. Do you guys know about the Wii? Maybe,
Yeah.
yeah, okay. So when it was first came out, it was like really popular, and then there was this radio station in the States. They held a contest. The contest was called hold your Wii, as in like your P, to win a Wii. Right, so this is what the contest was like. This is real. You can look it up. The contestants were made to drink water, okay, a lot of it, and they cannot go pee. That's the rule. Right? And if you're able to hold it, and you're the last person standing, then you win the Wii, like the video game console. Right? So this lady won the game, won the contest. She she was able to hold the water, and she went home with the Wii. But she died in that afternoon in her apartment. Why? Well, you can probably guess. Drinking excessive amount of water is going to make your blood really diluted, right? So what kind of solution is the blood in that case? Hyper, hypo, or iso? If you drink a lot of water. Which one is it, sorry?
Hyper.
All right, why don't we do a poll? Okay, I want to see if you guys actually understand what I'm saying. I am asking you, the person who drank a lot of water, she did not pee, she just hold it in, a lot of water. Is that gonna make her blood hypo, hyper, or iso? What do you think? Okay, please choose something and let me know. Okay, I have 9 answers. Anyone else? Okay. All right, looks like I failed to teach you guys. Okay, only half of you got it right? Okay. So listen. When you drink a lot of water, you're just talking about like regular water, right? Is there any salt in the water that you drink? Tell me.
No.
No, water is just water. There's no salt in it. There's no solute in it, right? There are no where's the note? There are no like red dots in it, right? So look, right? You drink a lot of water, and then you have your cell. The cell's gonna have its red dots, but the water, there's nothing, right? So what's gonna happen? They will come in. The water will come in because that's where the solutes are. So this is hypotonic. The cell's gonna swell up, right? You can remember hypotonic. Okay, this is the one, the big O. That means the cell's gonna swell up. All right? That's why the person died. Because she drank so much water that her cells, her red cells started to pop. Right? And there's no turning back from that, I told you. Make sense? Okay. So now that we have a basic understanding of things like diffusion and osmosis, we're gonna switch gear and talk about the type of transport across membrane. Okay, so this is an overview of the different kinds of transport we have. We're gonna redraw this thing later on. You can come back to it later and review it. Okay, but for the most part, the transport is separated into passive transport and active transport. Passive transport does not require cell energy. Okay? And it just happens spontaneously. It happens on its own. And that involves the movement of molecules from high concentration to low concentration. It just happens on its own. On the other hand, active transport requires energy in the form of ATP, which we've learned already. And it's going to be movement of molecules from low concentration to high concentration. It's the opposite. It's easier to visualize this by thinking about this analogy. If you are on top of a hill, the ball will roll down on its own, right? You give it a nudge and it's gonna go. No energy is required. This is easy, right? Whereas active transport is difficult because you have to go from low area to high area. You're going against the gradient. So you have to exert effort. You have to exert energy. Okay? So I'm gonna show you a bunch of transport, and I'm gonna tell you whether it's passive or active, and I'll try to describe it. And in the end, I will summarize it for you. All right. So first one is simple diffusion. This is passive. We learned about diffusion at the beginning of today's class, right? That's just the movement of particles from an area of high concentration to an area of low concentration. Okay? And this is going to work if the molecule is small. If it's small enough, it's going to be able to squeeze between the gaps in the cell membrane, and it's going to go in or go out. It's also good if it's hydrophobic. Right? Hydrophobic will have no problem crossing the phospholipid bilayer because much of the phospholipid bilayer is hydrophobic, right? Like those tails, right? The middle portion is hydrophobic. So things like steroid hormones, they can cross, no problem. Okay. And I don't know if I told, oh, I don't think I did. Never mind. Okay. I'll tell you another time. It was my other class. And even in non-polar molecules, if they're small, like CO2 and oxygen, they will be able to cross as well. Okay. Now don't think about it as, you know, always coming in. Whether it's in or out is determined by the concentration difference. Right? In this particular diagram, you have more of the red stuff outside than inside. That's why they're coming in. Right? If you have more on the inside than outside, then they will end up leaving. This is just a spontaneous process. It will continue to happen until the concentration are the same. Okay. When it's the same, we say it has reached equilibrium. Okay, so small molecules, hydrophobic ones, non-polar ones. Okay? That's passive. Sometimes the molecules are too big or they are charged and they're not able to cross the membrane by themselves. So they use the help of a membrane protein. Right? When that happens, we call it facilitated diffusion. Okay? Requires the help of a membrane protein. So we have two situations here. On the left side, we have what's called channel protein. They literally provide a tunnel for the things to pass through. Okay, so this is ideal for charged particles. If it's charged, it cannot cross the barrier by itself because of the phospholipid, because of the hydrophobic tails. So by having a tunnel, the charged particle can be hidden away from the hydrophobic tails, and it's able to cross.
Can you repeat it, please?
Sure. So these are charged particles. Okay, let's say Ca2+, Ca2+. Okay, the inside of the membrane is hydrophobic, right? Hydrophobic things and charged things don't mix together. Just like water and oil, they don't mix together, okay? So in order for the calcium to come into the cell, it has to go through this channel protein. The channel protein creates a tunnel for things to pass through. Literally, it's like the toilet paper roll here, okay? Like if you want to go through, right? There's something comes in, literally just pops right through like that, okay? So it shields the charged particle from the surrounding areas. Okay. It's like a door, right? Like you can't go through the wall. So let's put a door in the wall so you can pass through it. Understand? The other one here is called carrier protein. It's kind of like channel protein, but carrier protein is more like, you know, those things at the mall sometimes. What do you call them? Like a revolving door. Right? Like you go in and then you push and then you come out on the other side, right? Okay, so the carrier protein will grab onto one of these particles. It will change shape, and then it will end up opening on the other side, and that's how you move the particle across. Okay, so these are suitable for like big molecules. Okay, if it's bigger, it won't fit through the tunnel, so you gotta grab onto them and then physically move them over. Both of them does not require energy. Both of them are examples of passive transport. Okay, so this is just another picture that shows you that the carrier protein alternates between two shapes, two conformation. We learned about this word before. Moving molecules across the membrane as the shape of the protein changes. Okay, so it just grabs it, opens down, grabs another one, open up. Whereas the tunnel protein, there is no change in shape at all. It's just a hole. Okay, so things goes through and then comes out on the other end. Down their concentration gradient. Okay. So there are many factors that affect the membrane permeability. Okay, so membrane composition, for example. All right, we talk about how if you have more cholesterol or more saturated fatty acid, that's going to change the fluidity, right? Different cells have different membrane composition, and therefore they have different membrane permeability. Okay. Temperature is going to affect the membrane permeability as well, and that's what the lab is about. Okay. You will investigate how temperature affects the membrane permeability. Okay. There's going to be pH, ionic concentration, and whether the environment is hydrophilic or hydrophobic. So all these things are going to affect the membrane permeability. Okay, so your Beetroot lab is going to be done online, and you know, I can quickly show you. It's actually pretty straightforward. So you go to your eCentennial. That is totally the wrong class. Let me try that again. Okay, you go to eCentennial. I'll post a link for you to access it, okay? But it's under assessment and quizzes. It's not a quiz, okay? I'm just using the quiz platform to do the lab. So there is a Beetroot lab. It should be available now already. And it's going to be due on February 7th, which is next Wednesday, I believe. Yeah. Okay, it's going to due at midnight. Okay. So, I mean, you can start it. There are some stuff you can read here, but basically, there are some videos that you have to watch. It's me, okay? It's me doing the experiment. Or you're just gonna watch me do it. You have to watch it. If you don't watch it, you don't know what the lab is about, right? These are not questions you can just like, you know, randomly answer, right? And if you're stuck, there are additional help video. Like if you forgot what standardized variables are or whatever, right? Like you can click on the extra help video and then you can watch it. Even if you think you know the answer, just watch it. All right? Because like by watching it, it might make your answer better, right? Okay. So a lot of the answers that I need you to tell me are found in the videos. Okay, if you watch the video content and then the extra help video, you'll be able to find the answer. If not, it's gonna be in your class notes or in some of the lectures that we have done. Okay, you just have to use these resources to answer the question. Please do me a favor. Do not search the internet for the answers. I'm not telling you not to do it because, you know, first of all, I don't want you to be copying from the internet. But the problem is when you search stuff up on the internet, a lot of times you end up writing the wrong answers. Okay, because there's a lot of stuff on the internet, and sometimes they explain things in a way that's really complicated and it does not apply to what we are talking about. So if you copy from something like that, I will definitely know because it would seems weird and awkward, and the sentence will be, you know, about things that we haven't talked about. Right? So don't do it. Just watch the videos. I made it super simple for you. Okay, most of what you need is in the videos and in the class notes. The time limit is 90 minutes. That's plenty of time. Hour and a half to do this thing. Okay, so just find a quiet place and then do it. Right, so, you know, I'll start the quiz. Watch the video on background information. Okay, so like there's the video, right? Okay, and if you want to watch it on YouTube instead, you can click on the YouTube button. So there's like a watch on YouTube button, right? So this is some background information. Okay, you see here is me there. I used to have shorter hair. And then, you know, you can just watch the whole thing. Okay, I actually recorded this when I had I had COVID and I was quarantined in the basement away from my family and I had nothing to do. So I recorded these videos for you. So you can watch that. Okay. And then there's a question here is that the question should be in red so you cannot miss it. What is the purpose of this experiment? Okay, so this is what you do. You type in the answer. Okay, the purpose of this experiment is to investigate the effect of temperature on the beetroot membrane permeability. That's it. Okay, that's 1 point. I give you permission to copy this. Okay, take a picture. Just type me this exact answer. I'll give you 1 mark. Okay? Don't go crazy with the experiment. Don't write me a novel, please. Okay, just short and sweet and to the point. Okay, so there, copy it. I'm serious. You can copy it. I'm not going to be upset or anything. Okay, next. What is the name of the main pigment that creates a distinct, vibrant red color in beetroot? You don't know, right? I don't expect you to know. So watch the video. Oh, look, beetroot pigment. It actually tells you. That's what I mean, okay? You just have to watch the video. Don't go Google this. If you Google this, they will tell you something weird, and then I will know it's weird. And that's it. You just keep on doing it, doing it, right? Next page. And then it says, watch the following 3 videos before answering the question. All right. So again, if you want to watch a bigger, you just click on the YouTube button. But that is the lab. Like if you were in class, we will be doing this, but then I did it on your behalf, and you just watch it. Okay, so watch it, all of them. And then there is a question. What is independent variable in this experiment? Okay, and then you can type the answer here. But if you forgot what independent variable means, don't worry. There is another video here of me explaining what that means. Okay, so this is very straightforward, guys. Okay, it's like 10% of your final grade. You just have to do this whole thing. Keep track of the time in the top right corner. And when you're done, you submit it. That's it. That's your first lab. Does anybody have any question?
How many questions are there?
How many questions are there? Oh, I forgot. Sorry. Let me see. Let me start the quiz again. 20 questions. 20 questions. Okay. It sounds a lot, but
One attempt.
yeah, 1 attempt. Yeah, 1 attempt. It sounds like a lot, but it's not, right? Because most of the questions are just like, what is the purpose of this experiment? Okay, 19. It's. Are you guys worried about the time? 90 minutes is like not enough. You worry?
Like how many
Yes.
I
take
think
to
we're
watch the video?
Okay, so give me a time. What's a reasonable time? Tell me.
2 hours, Professor.
2 hours? Okay, sure. Hold on.
I don't know about others. If they...
Yeah, and oh, he's so agreeable. Let's ask for 3 hours, right? Hold on.
Video time limit
So
in
actually,
our
we need
class.
2 hours.
Yeah, sure, no problem. 2 hours, you got it. Okay, everybody happy now?
Thank you.
Yeah,
Thank
yeah,
you so much, Professor.
yeah. No worries.
Excuse
Okay.
me.
Yeah.
I want to ask, we just write down the answer of the question. We don't need to do the experiment.
That's right. You don't have to do the
in
experiment.
person.
Yeah, you don't have to go buy beetroot. You just watch me do it. Okay.
Okay.
Yeah.
Thank you.
That beetroot you saw in the video was actually supplied by your chemistry teacher. Okay. She grew it in her backyard and she had too many, so she gave me one to to film the video. Okay, so you guys can thank her for that. But anyway, that's the lab. Okay, it's available now, and you have until next Wednesday to hand it in.
So do we
okay?
have class on Monday and Wednesday? You were talking about no
Yeah,
class.
so I don't know about Monday yet. I'll see if I'm able to finish the lecture here. If I'll tell you at the end, but definitely no class on Wednesday. Okay, Wednesday you can just take the time to finish up the lab for me. Okay. All right. I think we should take a break. Okay. We'll return at 1:45. 1:45. All right. See you in a bit. If you have questions about the lab, feel free to type it in the chat, and I'll address them when I come back. Okay. See ya. All right, hello, it's me again. Let's keep going.
Sure.
Yeah, you had a question?
Yeah. You just gave us the hands up, which we can write the question and answer. The question number 8 and 9, I didn't get it.
Sorry, what's that?
That's hands out. You just not a hands out. The question you just gave us for the practice
Yeah, for the quiz.
of
Yeah, what?
this. I think it's like question number 8 and question number 9.
Question number 8 and 9 of
Yeah,
the lab.
not a lab. This chapter of remembering. Great question.
Sorry, which question 8 and 9 are you talking about? The study guide, is that
Yeah, membrane transport is study.
this one? Number
Yeah,
8 and
yeah.
9? Oh, yeah. So the answer is it's on the study guide answers, right? Like, because I don't have time in class to take up all the questions, right, in the study guide. So as I was saying, hold on. At the end of each class, you should check the eCentennial, and it will tell you the lecture archives, class notes, and study guide answers have been updated. You just click on that, and then that's where you can find the answers, right? Okay, so like I haven't given you the lecture 4 answers yet because we haven't finished the lecture, right? Okay, but like after class, I'll turn it on, then you'll be able to see it. All right? Okay. So here, passive membrane transport processes include consumption of ATP. That's not true. No ATP are needed for passive transport. Movement of substance down its concentration gradient. That's correct. Down concentration gradient means from high to low. The use of transport protein while moving substance from area of low to high. Nope. Low to high is active transport. Movement of water from an area of low concentration to an area of high concentration. That is also not true. Okay. So it's just B. Okay, let's try this one. See if you learn anything before the break. Go ahead. Okay, it has been more than a minute. Anyone else want to lock in the answer before I close the poll? All right. So hypertonic, if you look at the notes we took earlier today, that's when the cell is, the water is basically leaving the cell, right? So net movement of water is out of the cell, which will cause it to shrink. And the reason why the water is leaving is because the concentration is higher outside compared to the inside. So A, C, and E are all correct. Okay, so now let's turn our attention to active transport. Active transport. They require ATP, and it's moving things from low concentration to high concentration. First example is protein pump. Protein pumps is exactly what it sounds like. These are membrane proteins that uses ATP to pump things against their concentration gradient. So going from low to high concentration. And it's useful for keeping unwanted things out of the cell, as well as bringing certain things in. An example of that would be a proton pump. You know you have acid in your stomach, right? So to create that acid, the cells in the stomach lining, they have these proton pumps. They keep on pumping H+ into the acid, into the stomach acid to keep it acidic. People who have really bad heartburn, sometimes the doctor will give them PPI, which is proton pump inhibitor. By blocking these proton pumps, you make less acid, and it's gonna make your heartburn better. Another example is sodium-potassium pump. Sodium-potassium pump is important for normal cell function. And we will learn a lot more about the sodium-potassium pump next semester. That basically it works by moving 3 sodium out of the cell in exchange for 2 potassium. Just stay with me. If we keep on pumping sodium out of the cell, it means eventually you will have high sodium on the outside and low sodium on the inside, right? So in order for this process to keep on happening, you have to use ATP because you are pumping against the concentration gradient, right? The sodium actually wants to come back in, but you keep on pushing them out with the pump. All right, that's active transport. Same thing for the potassium, the K, right? If you keep on bringing potassium in, it's going to be high concentration inside and low concentration outside. And you're going against its concentration gradient as well. So in these situations where you're going from low to high, you are using ATP. Another example of active transport is called bulk transport. Bulk means a lot of it, right? Or really big, right? So things that are bulky, such as an entire bacterium or large particles like cholesterols, they have to move across the plasma membrane through bulk transport. And there are 2 types, endocytosis and exocytosis. In endocytosis, you are bringing things in. So this here, this bottom picture, that's endocytosis. Now you have all these pink dots that you want to bring in. So the cell membrane is going to create a little cave, and eventually it's going to pinches off like that, and then you have a vesicle. Okay? Where is the vesicle going to go? Does anybody remember from lecture 3? If I want to like process these purple dots that I just brought into the cell, if I want to break them down, where should I take it? Which organelle helps break down particles?
Lysosome.
That's right, lysosome. Thank you. If you didn't know that, then maybe there is some studying that you need to do, right? So this may go to the lysosome and gets processed. All right. The opposite of endocytosis is exocytosis. So if you have a bunch of stuff that you need to get rid of, or maybe you just created a bunch of proteins, right? You just finished packaging them from the Golgi and you want to send it out of the cell. So the vesicle is going to fuse with the membrane and release the content. Okay, so either way, this is for moving large amount of material into or out of the cell. When it comes to endocytosis, bringing things in, there are 3 types. Okay. Number 1 is called phagocytosis. That is if you want to bring in particles, large pieces of particles that you want to bring into the cell. For example, your white blood cell, they can eat up a bacteria. That's called phagocytosis. They could actually eat up cells that are being infected as well. Okay, so that's cell eating. That's what this means. Okay. Pinocytosis is cell drinking. Cell drinking. And instead of taking in particles, you are mostly taking in fluid. You're mostly taking in fluid. The human egg can take up nutrients from the environment through pinocytosis. If you're bringing in solid, phagocytosis. If you're bringing in liquid, pinocytosis. And the last one is receptor-mediated endocytosis. Okay. This is very similar to phagocytosis, but it's more specific about what it is taking in. Okay. So in order to bring the thing in, it must first bind to a particular receptor. Remember we talked about receptor as a membrane protein, right? So there are these things with certain shape, and if the shape fits, that's gonna bring them in. Yes, Aminette, you had a question? Let's see. Oh no, okay, that's fine. So cholesterols are absorbed this way in the liver. And here are 3 pictures that shows you side by side how they are different. Okay, so phagocytosis, you bring in the solid. Okay. Don't worry about these like pseudopodium, okay? Pinocytosis is you bring in the liquid, which you might bring in some dissolved substances as well. And for receptor-mediated endocytosis, you must first bind to a special receptor. And the receptor will cause the cell membrane to kind of fall into this vesicle and bring it in. Okay, so all of these are endocytosis. All of these are about bringing things into the cell. But what you bring in and how you bring it in is what distinguishes them. Okay, there are a few concept check questions. Let's go through them together. I'm not gonna do the poll because it looks like you guys are not really participating. So it's okay. I'll just tell you the answer. Which of the following can cross the cell membrane easiest? Okay, so we want something small, right? So if it says large, then that's not the case. Okay, hydrophilic stuff also have trouble crossing it. Okay, charge also have problem crossing it. Remember, if it's charged, the best way to bring them in is through the channel proteins, right? The tunnels, right? Bacterium is really big. For those of you who don't know, bacterium is it's one. Bacteria, it's many. So the correct answer is this. If it's small and it's hydrophobic, we can bring them in easily. A patient has had a serious accident and lost a lot of blood. To replenish body fluids, distilled water equal to the volume of blood loss is transferred directly into one of his veins. What will be the most probable result of this transfusion? Anybody want to tell me what happens if you put distilled water into the veins? Is it going to help this person who has lost a lot of blood?
No, I think it needs saline water, right?
That's right. And what happens if I give them distilled water? What's going to happen to him?
They don't have any sodium in their blood.
That's right. So there's no sodium, no electrolyte, right? So he's going to experience his cell is going to be in a hypotonic, HYPO, right? Hypotonic scenario, and the cell might start to burst, right? So this is a bad idea. We should not be giving him distilled water. Okay, so the answer is the probable result is that his cell is going to burst because the distilled water creates a hypotonic environment. I'll read this to you. 5 bags constructed from a semi-permeable membrane that is impermeable to sucrose. That's just a kind of sugar, right? Your kitchen sugar. Were filled with various concentration of sucrose and then placed in a separate beakers containing an initial concentration of 0.6 m sucrose solution. It's okay if you don't understand what m is. You learn that in chemistry later. But this is just a way to measure the concentration, right? 0.6. So at 10 minutes interval, the bag were weighted and the percent change in mass of each bag was graphed. Okay, so like we have 5 bags here. Which line represents the bag that contains the solution isotonic to 0.6 molar solution? Right, the key word here is isotonic. How is the volume going to change if it's isotonic? Is it gonna change?
No.
No. So it's gonna be this flat line right here. Okay, no change in volume, right? This one is hypotonic. And this one is hypertonic. Okay. I guess both of them are hypotonic. Both of these are hypertonic. Okay. But linear, like, it doesn't matter actually. Yeah, that's just it is. Okay. Last one. This one is important for one of the lab questions. Okay. So I want to know what you guys think. Go ahead. Okay, thank you. Much more response this time. So this is a trick question, okay? I know what you guys thinking. You see hypotonic and then you choose swell and burst, right? But here's the thing. It says after freezing and thawing. That's the key thing here. What happens if you freeze water? You fill up a water bottle and then you freeze it. Like, yeah, sure, it turns into ice, but then like, can you describe to me what's gonna happen to your water bottle? Anybody?
It swells up a little bit.
Yeah, it swells up a little bit, right? Expands, right? Okay. When water freezes, it expands. Right? So what happens is when you freeze the animal cell, the ice crystals actually puncture the cell membrane. Cell membrane damaged by freezing. Okay, because the water expands when it freezes, and that all combined with the ice crystals, it damages the cell membrane. So after you thaw it, the cell membrane is already full of holes. So it doesn't matter. It doesn't matter whether you put it in hypotonic, hypertonic solution, it's full of holes. There's no more regulation. Things just goes in and out with no control, right? So no change in size. Okay. Please keep this in mind when you do one of the questions in the lab. By the way, have you guys learned how to graph?
Sorry, Vincent, it happens to plant cells as well, this one?
Yes and yes and no. Okay, like you might wonder, hey, how come the plants in the winter, they don't just like, die, right? Like, well, some plants can produce some antifreeze naturally, so it prevents this process from damaging the cells. Right? But if you take a piece of like a vegetable, if you buy some green vegetables like spinach or kale, and you put it in the freezer, and then you take it out, don't put it in the water, just put the whole leaf in the freezer, and then you take it out after, you will notice it's like really watery, right? Like, where does the water come from? Like, you never really soak it in anything, right? That's just the freezing damages all the cell, and all the water content within the cell now leaks out because the cell membrane is not intact anymore, right?
Okay, thank you.
Yeah. Did you guys learn how to graph in your computer class yet?
Yes.
Yes. So sorry, I didn't quite catch that. Yes.
Yes.
Yeah. Okay. So you have to do a graph for the data analysis in the lab. Okay. So if you forgot, I think I also have a video that shows you how to do it using Google Sheets. Okay. If you know how to do it on Excel and you have Excel, that's fine as well. All right. That's it. That's it for today. Please, I think we're gonna have a class on Monday, okay? Because there is no class on Family Day, right? So I want to be able to finish teaching everything before your actual test. So please come on Monday. We will we'll do a quick review of lecture 4, and then we will start on lecture 5 a little bit, okay? But no class on Wednesday for sure. All right. Okay. Thank you.
Thank you.
Thank
Bye.
you.
Excuse me, Professor.
Yeah. Go ahead.